Bismillahirrahmanirrahim. Today we will discuss complement system, and this is the end result of the complement system. End of the lecture we will understand this picture clearly. What is complement system? Composition of complement. Complement describe a system of about 20 proteins, many of which are enzyme precursor. Enzyme precursor means they are prone to, when activated, they become active enzyme, and precursor means they are in inactive form. The principal actor in this system are 11 proteins designated C1 through C9, B and D. C1 to C9 are given the numerical name, and B and D are given the alphabetic name. Here it is written that there are 20 protein in this system, but in some book it is written that there are 30 proteins in this system, or maybe more. All these are present normally among the plasma protein in the blood, as well as among the protein that leak out of the capillary into the tissue space. We know that in the blood we have plasma, in plasma we have protein, multiple proteins. So these proteins are present mixed with the plasma protein, and when the blood leaks from the circulatory system into the surrounding tissue or the extracellular fluid, these proteins also leak with these plasma protein into the extracellular fluid. The enzyme precursors are normally inactive. As I told you, in active form they are called enzyme precursor, but they can be activated mainly by a pathway which is called classical pathway. Another pathway is also there, which is called alternate pathway. So by this pathway the complement system is activated. Production of complements proteins. Liver is the main organ which is responsible for the production of these proteins, and in the first 13 weeks of intrauterine life, these proteins appear in the plasma of the infant. Another organ which is responsible for the production of C1 protein is mucosa of the gastrointestinal tract. Functions responsible for certain aspect of immune response and inflammatory response. They take part in the immune activity and also in the inflammatory activity. The activation occurs due to antigen and antibody reaction. When antigen and antibody react with each other and make antigen-antibody complex, this system is activated. Or endotoxin of the bacteria or capsule series of protein activate this system sequentially, in sequence. This is not activated haphazardly, but it is activated in a sequential manner. And inactivation or inhibition of these proteins are caused by inhibitor substances which are present in the plasma. When there is no abnormality, disease in the body, no antigen in the body, no bacteria in the body, these proteins are present in inactive form, and their lifespan is short-lived. They are short-lived. They do beneficial activity for the body, or they may be also harmful to the host. Complement activation pathways. One is the classic pathway, the other is alternate. This is the alternate pathway, and it starts with the antigen antibody reaction and end in a attacking structure which attack the membrane and the alternate pathway, in which antigen and antibody complex is responsible for the activation of classical pathway. While alternate pathway, bacteria is responsible for activation. Composition of complement protein. Each complement protein is composed of two parts, A and B. When it is in inactive status, it has two parts, B and A, or A and B part. The B part is the large part and the A part is the small portion of the complement proteins. A is small portion, and when activated, detach and diffuse into the plasma. This complement protein is activated. The small portion, A portion, is detached from the B and diffuse into the plasma. B is large portion and bind to other proteins on the cascade and activate them. This protein, this is activated part of the complement protein and take part in the cascade reaction of the complement system and combine to the other protein of the complement and activate them. Here you can see a bar, and this is a complement protein 1. When bar is put on it means this protein is activated. Remember, B is for binding or baby and A is for apple. When the baby is irritated and when it move its head, the apple is dropped from its head and then it goes to the plasma. Classical pathway. How the classical pathway is activated. The classical pathway of complement is activated by antigen-antibody complex and two immunoglobulin, the IgM or IgA, is responsible for the activation of classical pathway. Fc portion of the antibody form a binding site for C1q. When there is antigen-antibody reaction, the portion of complement C1q attached to the Fc portion of the antibody. This Q portion of the C protein is attached to the Fc portion, and when this attachment occurs, the complement activation system started. Then the numerical sequence of complement factor activation in the classical pathway is: first, C1 react with the Fc portion of the antibody. When this reaction occurs, the C1 activates C4. Remember, not the C3. The C4 activates the C2. Then the 3, 5, 6, 7, 8, and 9. The rest. Before 3, the C is activated. Remember this, that when the classical pathway is activated, the C protein activates the C4 first. This is the sequence of activation. Classical pathways. First, the C1 is activated or it is attached with the tail of the antibody with the Fc portion and it activates C4. Then C4 activates C2. C2 and C4 activate C3. C3 can be also activated directly by the alternate pathway. C4 then activates C5, 6, 7, 8, 9. And in the last, a membrane damage protein form which cause damage to the membrane and burst the bacteria. This is antibody which has a long arm and a short arm, the variable portion and the non-variable portion. Attachment part for antibody is present on the permanent portion of the antibody or on the long arm of the antibody, and this is called Fc portion, and this is the complement activation pathway. Here in this large picture you can see that this is a target cell or pathogen cell. It contains antigen. On the surface of it there are present antigen, and when antibody attached to it, it exposes the Fc portion on the tail of the antibody to which C1 protein is attached. When C1 protein is attached, it activates C4 and C2. This C4 is broken down into two portions. One is the small portion and the apple portion, which diffuse into the plasma, and the activated portion bind with the C2 activated portion and make a complex. This complex is attached with the target cell. This target cell contains antibody, the C4b portion and the C2b portion. This complex is called C3 convertase. So this C3 convertase act on C3 and activate it. This C3 convertase then goes and attach with the C4b and C2b. This is attached with them this and make a complex. The C3b, C4b, and C2a. They make a complex which is called C5 convertase. And the C3b has another capability. It divides multi-time and it deposits on the surface of the cell. This cell, the target cell, its activated portion is also combined with the C2b and C4b and also on the surface of membrane of the target cell. This C5 convertase activate C5 protein and break it into a small portion A and activated B portion. This goes into the plasma and the C5, this is the 3, the 2 and the 4 complex. When 5 is combined with it, it make a convertase or it activates C6, C7 and C8. When this complex is formed, it activates complement 9 and multiple protein of complement 9 join together in a cylindrical fashion with each other and make a structure which is called membrane attacking complex. So from up to here it is activation portion of the complement. From C8 beyond, attacking portion is then formed and it physically invade the cell membrane and make a hole in the bacterial cell membrane and expose the cell membrane to the external environment. Content inside it is expelled outside and the content outside of the cell membrane diffuse inside and it swell up and burst the cell membrane of the bacteria and this way kill the bacteria. There is another pathway which is called MBL. MBL is mannose binding lecithin protein. By this is a complement path 3 and it is resemble classical pathway. By this pathway also the complement is activated and the rest of the stages are same as in the classical pathway. The alternate pathway is activated by spontaneous hydrolysis. The C3 component is spontaneously hydrolyzed by factor B, protein B and D. The protein B activates C3 and in the presence of D it is activated into C convertase which activate the C protein. So C is from inactive phase, it is converted into active status. This active C3 then act on the C5, this is C5 convertase and ultimately the rest of the pathway is started and it then rupture the membrane of the invader organism. Here the C3 has the capability to attach to C4b and C2b, but it also has the capability to deposit on the cell membrane, and when it is deposited on the cell membrane, this cell is exposed to the CR3, cell membrane 3 protein. When the C3 deposit on the surface of this cell, it exposes this to the phagocytic cells, and this why it is called opsonine. The C3 protein is also called opsonine. When the C3 is activated by the bacterial endotoxin or capsule membrane, then the rest of the pathway is the same to the membrane attacking portion. The classical pathway is initiated by an antigen-antibody reaction. A specific reactive site on the constant portion of antibody become uncoiled or activated, and this in turn bind directly with the C1 molecule of the complement system, setting into the motion of cascade of sequential reaction. Binding with activation of the proenzyme C1 itself, the C1 enzyme that are formed then activated successfully, increasing quantities of enzyme in a later stage of the system so that from a small binding an extremely large amplified reaction is developed. So here this is the pathway C1 activation to C4 and C2 is activated. A bar is present. This means this is an activated portion. But in this picture, the most important thing is when these undetached portion of C4 diffuse into the plasma and the C3A portion when it diffuse into the plasma and C5A portion where they perform some activity in the body or in the plasma. The C4 activate mast cell. You can see that the C4 small portion activate the mast cell. Also the C3 activate the mast cell. The C5 activate the basophilic cell and that release histamine, bradykinin and anaphylactic substances. That's why this portion of the C protein is called anaphylactoxin. This is the antibody and this is the Fc portion of the antibody and this C1 is actually attached to this portion. C1 exists in blood serum as molecular complex containing one molecule of C1, two molecule of C3 and two molecule of CS. So C1 protein is divided again into three portions. It contains one molecule of C1, two molecule of R and two molecule of S. QRX complex, you may remember it in the ECG. The constant region of antibody contain a binding site for C1q. A single molecule of IgM is enough to inactivate the pathway. Here you can see this is the Fc portion. Here a binding site is present on the Fc portion. This is when there is no antigen-antibody reaction. This portion is remain hidden or this is covered. But when antigen-antibody reaction occur, this portion is uncovered or activated. And this attract the C1q. As I told you, the C protein contains three portions. So the Q portion attached to the area and it activates the complement or the classical pathway. Here you can see that this is the C1 protein. You can see that here we have two molecules of S, two molecules of R and one molecule of Q. This is the C1 protein. The C1 protein attached to the antibody IgG or IgM to the tail. There is a site for its attachment. It is normally called, but when there is antigen-antibody reaction, when some antigen is attached to this antibody, the tail exposes the area to which CQ molecule is attached of the C1 protein. This causes activation of the classical pathway. When this attachment occurs, the S component, which has two molecules on one side, it's break down or activate the C4 into two portions, the C4A and C4B. This bring the C4 or attract the C4 molecule and then cleave it into two portions, A and B. The B is the active or the binding portion of the C4 molecule. It attached to the cell membrane and the C4 diffuse into the plasma. On the other side, it activates C2. It also brings the C2 molecule attract and then it cleave it into C2A and C2B. The B portion actually then come and attach to the C4 molecule. This make a complex. C2 and C4 make a complex. This is C4 and this is C2. They make a complex on the surface of the cell membrane. When the C2 and C4 combine with each other, they attract C3 and cleave it or break it into two portions, C3A and C3B. This C3B done two jobs. One, it multiply multi-time or divide multiple time and produce many molecules of C3B, which are active molecule of the C protein, and these molecules decorate or deposit on the cell membrane surface. And also this C3B attached to the C4, which actually make a C convertase for the protein C. C3 is the most abundant protein of the component complex, and its composition is 1.3 milligram per milliliter. Opsonization: C4B and C2B cut C3 into two major fragments. C3b, which bind covalently to glycoproteins secreted across the cell surface. Macrophages and neutrophils have receptor for C3b. The neutrophil and macrophages have receptor for the C3b. This is actually deposited or decorated on the cell membrane. On the other hand, here the neutrophil and macrophages are present and they have receptor for this C3b, and then they bind to the C3b coated cell or particle preparatory to the phagocytosis. So when these C3b are present on the surface of any cell, they are attacked by the phagocytic cell and phagocyte them. This effect qualifies C3b as an opsonine. Opsonine means they bring the bacteria and the phagocytic cell near to each other. So this is called opsonine. They bring it to close to each other. The C3b bring the phagocytic cell close to the bacteria. The C3a, this is small fragment, is released into the surrounding fluid. This C3, when cleaved, this portion is released into the surrounding tissue. It can bind to receptor on basophil and mast cell, trigger them to release their vasoconstrictive content, histamine, because of this role of this material in the anaphylaxis. C3 is called anaphylotoxin. It causes anaphylaxis. The C3A portion, when it binds to the basophilic cell, it releases histamine and bradykinin and it causes anaphylaxis. Some of the C3 bind to the molecule of C5, expose them to cleave by C4B2, which is C3-5 convertase. This is again the classical and the alternate pathway. Now we'll discuss the alternate pathway. The alternate pathway initiated by bacterial endotoxin, polysaccharide capsule, aggregate of IgE and properdine. Properdine is the only gene of the complement system that is encoded on the X chromosome. Properdine stabilizes the alternate pathway C3 and C5 convertase by it starts as C3, then C5, C6, C7, C8, and C9. The complement complex C1, C4, C2 are bypassed. In classical pathway, remember the C1, C4, and C2 pathway is bypassed and it is directly activated from the C3 by these endotoxin or capsule of the bacteria. Antibodies are not required to initiate or activate this pathway. This pathway provides a mean of non-specific immunity. Here you can see, as I told you in the lecture in the beginning, that what is the meaning of this picture. Here a barrel-shaped form. This is membrane attacking portion by the alternate pathway, and by the classical pathway the protein 9 make a cylindrical attach with these 7, 6 and 8. These make a cylindrical structure which attach with each other and make an membrane attacking a component which physically make a hole in the membrane and expose the bacteria inside to the external environment or to the extracellular fluid, which gush into inside of the cell, and the bacterial cell components are leaked out, and in this way the membrane is burst out. So this was the picture which I was given in the initial slides. Now you can understand what this picture means and how by the bacterial wall is destroyed by this